The Jerry Powell podcast is supported by listeners like you who've either spread the word on social media, left reviews on podcasting apps, or have clicked on our donate button on the Jerry Powell website so we can continue to fund each one of these episodes. Welcome to the Jerry Powell podcast. This is Eric Wadera.
This is Alex Smith.
And Alex, I'm going to love this one. Who do we have with us today?
This one won't be controversial at all. We're delighted to welcome back Jason Karlawish, who's professor of medicine and co-director of the Penn Memory Center at the University of Pennsylvania, Perelman School of Medicine. Welcome back to the Jerry Powell podcast, Jason.
Great to be back, Alex and Eric.
And we're delighted to welcome back Ken Covinsky, who's professor of medicine and geriatrics at UCSF. Welcome back, Ken.
It's an honor and a privilege to be back.
So today's podcast. So we've had Jason on before, talking about both dementia, his book, which we'll have links to in the show notes, and also about Aducanumab. But this podcast is not on Aducanumab. It's on lecanemab, which was just published in the New England Journal, anti-amyloid drug for dementia, Alzheimer's disease. Before we go into that topic, which we got a lot of ground to cover, Jason, I think you got a song request for Alex.
Alex, the times, they are changing.
Great choice. Pay close attention to the second verse lyrics. I think they're appropriate. Come gather around people wherever you roam and admit that the waters around you have grown and accept it that soon you'll be drenched to the bone. If your time to you is worth saving, then you better start swimming, 'cause you'll sink like a stone, for the times they are a-changin'. Come writers and critics who prophesize with your pen, and keep your eyes wide, the chance won't come again. And don't speak too soon, for the wheel's still in spin. There's no telling who that it's naming, for the loser now will be later to win, for the times they are a-changin'.
Bravo.
Very apropos.
Brilliant. The other title I wanted, which apparently was already co-opted by Shelley Morrison, was We Won't Get Fooled Again by The Who.
That is awesome. Isn't it cool that like Bob Dylan won a Nobel Prize for Literature for lyrics?
Very cool. Incredible songwriter.
Yeah.
Poet, yeah.
So a couple of weeks ago, we did a podcast on the need for cultural transformation around dementia care. We didn't bring up, actually, we touched upon lecanemab and anti-amyloid antibodies, but really thinking about other ways we can transform dementia care. This podcast, ultimately, we want to get to the question, should the field of geriatrics get on board with lecanemab and these anti-amyloid drugs? Because there's been a lot of pushback from geriatricians, including our national organization, American Geriatric Society, pushing back against at least Aducanumab. The question is, should we have that same take on this new drug? So let's start off from the beginning. Jason, what the heck is lecanemab? Am I pronouncing that right? I probably should know how to pronounce that.
It's a French version, right, of Aducanumab?
Exactly. Maybe. No, c'est très différent. Yeah, no, Aducanumab remains the dumpster fire. Lecanemab is, well, let's just say this, its study maybe says it all. It's Clarity. It was the name of their clinical trial, the Clarity study. Lecanemab is an anti-amyloid antibody. It's delivered by intravenous infusion every 2 weeks. It targets soluble amyloid species in the amyloid cascade. And it was studied in Clarity AD, phase 3, 18-month-long, single-dose, 50-50 drug versus placebo clinical trial, enrolling people with mild cognitive impairment, mild stage dementia, who had elevated amyloid by PET scan. That's the guts of the study, and that's the drug.
So it seems to really, again, another drug focused on the amyloid hypothesis. Do we know if it does anything to any other of these biomarkers like tau?
Yeah, so it is an anti-amyloid drug, and one key message with anti-amyloid drugs is they're not all the same. The amyloid cascade starts with the protein sitting benignly in the cell wall, then something happens, breaks off, forms fragments, fragments form fibrils form oligomers, and so on onto the plaques. And depending on the drug, bapineuzumab, solanezumab, they actually target different points along those protein malformations, just the way the antibodies are designed. So this one goes after, as I say, the soluble fragments. Aducanumab goes along, I don't know, the protofibrils or something. So they all slightly differ there. And this one lowered amyloid, as confirmed by PET scan. I mean, fairly, notable lowering on the centiloid measure of amyloid below the cutoff considered to move from not elevated to elevated. The secondary data on biomarkers, namely the tau data, the hyperphosphorylated tau that correlates with neurodegeneration, that also showed changes of decrease that would be befitting of a reduction in tau level, therefore a reduction in neurodegeneration. So it moved amyloid endpoints, as these drugs generally do, but more importantly, I kind of think it moved tau measures as well.
So let's dive into, so we're going to start off with the facts, the things that we know. And the great thing is, as opposed to aducanumab, the drug company actually published their studies first before going to the FDA, before having to, sift through, loads of webpage data from the FDA. We can actually pull up a New England Journal study that gives us some information about the efficacy and safety of this drug. Can we start off with what did they do in this study, this Clarity study? Who was enrolled?
Well, I'll continue on. As I say, people, adults with either mild cognitive impairment, so cognitive impairment that's detectable but causing inefficiencies in daily life, or mild stage dementia, meaning some disability. Mini-mental cutoff, I forget precisely, but I think it was low 20s was the cutoff, the lower cutoff. You had to have a definite impairment on a memory, a pretty intensive measure of memory called the logical memory test. And as I say, had to have evidence of elevated amyloid by a PET scan. Met those criteria, got in. Am I missing anything, Ken, or anyone?
That seems right. So is it fair to say, Jason, by and large, people who don't have Alzheimer's yet, or maybe in some cases ever, and people who, this is a population by and large, are still living at home, they're still capable of some independence, maybe even a lot of independence. Pre-nursing home kind of.
Oh, yeah. So this is a group of people that have some inefficiencies or impairments in higher level IADLs. So someone's got to do the finances, but they're still doing the cooking, et cetera. No troubles with BADLs stereotypically. Yeah.
One other point I'd add is in the aducanumab study, I believe you had to have the PET scan. Here you had a PET scan or CSF measurement of A-beta.
That is correct, yes.
Which may have implications as far as access to these drugs because PET scans are rare.
CMS does not cover PET scans as of yet. They're going to relook at that decision in a week or two, whether they'll PET scans.
And it sounds like this drug does a really good job of taking away amyloid. Is that right?
That's right. There's no contesting that. As did aducanumab, as did many of these drugs. But yes, the reduction of amyloid was well documented and very clear. Yeah.
And how well did it do with what do we know as far as the data around improving cognitive function and function in general?
So it didn't improve. These are diseases, that are degenerative. So the trajectory over time is loss of cognitive function and performance on activities of daily living, instrumental and basic. So the measure here was a composite measure called the Clinical Dementia Rating, or CDR. It's a measure that's used in research only. I don't think it has ever or will ever be used in clinical practice. And it's a composite measure that rates someone's performance on a sort of omnibus concepts of cognition, memory, judgment, decision-making, and then omnibus concepts of function like home and hobbies, personal care. And that was the primary endpoint. And is a very quirky way to look at the CDR, which is instead of the way it was designed, 0.5, 1, 2, 3, normal, very mild, moderate, severe dementia, you can look at the sum of boxes, which is essentially taking the subscores that make up those 0.5, 1, 2, 3, and treating them as subscores or the boxes, which is a quirk of the instrument. And so what they looked at was a difference in the CDR sum of boxes between decline in placebo versus the decline in drug group. That was the primary and single primary endpoint for the study, which has become the primary endpoint for a lot of these studies.
So Jason, just to make sure I have my head around this right, so you basically have 6 pieces of what people consider part of the dementia syndrome, 6 impacts, like ranging from memory to orientation to ability to do activities. And they're each scored on a value of like where 0 is normal, half is mildly, subtly abnormal. 1 is definitely abnormal, but mild, all the way to 3 to very abnormal. So you end up with, when we're sort of like, when we get to looking at this sum, we're looking at basically 3 times 18. 6. So we're looking at 18 points.
That's right. The boxes range from 0 to 18. And that's the scale. In this group of patients, the range of their CDR sum of boxes was about 0 to 6 with a mean median of 3. So that reiterates the question you asked there, Alex. These were people with mild disease. Their sum of boxes was stacked up at the mild stage, of severity. Even though the scale does range to 18, this was not a group of people with a median of 9, which would describe a group of people with moderate to severe as well as mild. This was very stacked in the mild stage. Yeah.
So scattering of mild to moderate deficits basically gets you to a 3.
A CDR global of 3?
Yeah, a total of 3. Yeah.
A total of 3 is severe stage dementia. That would be people with probably, I would crosswalk to a mini medal of about 12 or 10, in a well-characterized Alzheimer's population. In contrast, these people's global CDR was either 0.5 or 1. So again, this was a mild stage group of people. 0.5 typically describes MCI, although some people with 0.5 are just called having dementia. 1 generally describes someone with mild stage dementia. And this was a group of 0.5 or 1, or a sum of boxes score from about 0 to 6. Again, mean was about 3.5.
Okay, yeah, I was thinking more of the mean. So you get to this mean with a scattering of mild. It's like usually a number of mild impairments that I mean, I don't want to say they're mild because they matter a lot, but patients are experiencing a number of abnormalities, none of which are severe, but in aggregate might be bothering them a lot.
That would be correct. Yeah, I mean, they need caregivers, yes, for monitoring, supervision, and some assistance. Yeah, that's what I would call them.
And I can imagine one of the challenges is when everybody's skewed towards one side, it may be hard to see a difference. So when we think about that, can we even say what a minimally clinically significant change is, given that we're starting on one end of the spectrum?
Well, the theology is that a change on the CDR sum of boxes of, I think, about 0.3 something is considered to be significant. And I kind of listen to that and I say, fine. I think in this study and all these Alzheimer's studies, you have to have a primary endpoint because you're going to spend your power on that, and your alpha. But there are also secondaries that you want to look at. And ideally, you'd like to see all your primary and secondaries line up because it kind of points you in the direction that there's something going on here. So on the CDR sum of boxes, the placebo group didn't decline very fast in this group, as we'd expected, but the drug group declined slower than the placebo group. And if you do, there are various machinations. And I said that the way they analyzed the data, which is entirely appropriate intention to treat, showed an overall mean difference of 0.45 on the CDR sum of boxes between drug and placebo. That's the effect size.
And like you said, on all the secondary outcomes, they also showed similar kind of benefit. Is that right?
That's right. So they measured, again, you could debate the particulars of each measure, but they measured a series of standard measures of cognition, instrumental activities of daily living, a kind of a composite measure of cognition, instrumental activities of daily living. And on all those measures of cognition and function, the same results were obtained, which was people on drug did not decline with the same rate as people on placebo.
So how does that, like Eric, you used the term, you used a rather wonky term, was it minimally clinically important difference? And I don't know, would a good lay way of saying that be, like, okay, we see a number, but doesn't matter. So what difference matters? This is like in studies of where you're looking at these kind of difficult to measure things, you get a lot of debates on what that number is. I would just sort of point out my read of like, it's all over the map what people would say matters on this test. People who do drug trials seem to like suggest that the number is lower. People who are less into drug trials seem to say the number is higher. So I've seen numbers ranging all the way from like a change of 0.3, which this study would have met. But I've seen a number of other people suggest, well, the real number is 1. That's a number. We can't answer that question. It's an opinion and it's a value judgment. But, part of where, like, when we debate this, a lot of this really hinges on what really matters and would a patient or a family see something or feel better because of this change.
Yeah. It's funny because, dementia literature is ripe with statistically significant improvements, but clinically questionable. You look at the Ariceps of the world, cholinesterase inhibitors, memantine, it keeps on coming up. It's statistically significant. How important is it clinically? I think we're stuck in that same rut right now. Why do you think that is? Why do we keep on falling into this argument, which I would say it's an argument right now. I think you can make a case either way.
Yeah. So, we are. I'm not in a rut because if FDA approves the drug, and I think they probably will, unless there's some data that's going to come out that when reanalysis shows, but FDA will approve the drug. If they approve the drug, I'll prescribe it at the Penn Memory Center. It's going to be an interesting discussion because the drug, and we'll talk about it, does have risks. And I think there's some subgroups that need to be thought carefully about. But on balance, I think it crosses the threshold of being a drug that's efficacious, albeit a drug with risks. Really, for a drug like this, the final proof of its value is not known right now. That is, if you stay on it for, say, 2 years or more, what's its effect on your rate of progression? And from a societal perspective, what's that summative impact on the cumulative time spent caregiving, the cost of caregiving, et cetera? We just don't know that answer, but that's what we need to know.
Well,
because
let me
that's
ask
what
you about
matters
that.
in this disease.
So when I look at the graphs in the New England Journal paper, it looks like most of the change happens between 3 and 9 months. And then the slopes start looking the same between the placebo group and the lecanemab group. Suggesting, again, just it's eyeballing. Potentially suggesting to me is that, you get rid of some amyloid, maybe there's some moderate clinical benefit. That's a word that they use. They like the word moderate, I think, in the New England Journal paper.
Yeah. Well, they struck that from a substance. It was both in and then out. Yeah.
Oh, was it?
Yeah. It was put in the final version, but in the previous draft, it was moderate, was not there.
I imagine that was a great source of debate and compromise. I might have opted for minimal to mild, but one can debate that.
How much should we look at the trend lines? Like that they're after about 6 months to 9 months of treatment, I don't see them separating much more. Suggesting that 2 or 3 years later, are we really going to see any change, or have we just changed it this is the maximal effect?
So what you want to see, and we'll know this, is they then, when the Clarity AD study ended, randomized phase, those who had been randomized blinded to placebo were put on drug, and those on drug remained on drug. And what you want to see is once people who had been on 18 months of placebo are on drug, what happens to their curve? Do they jump up to the other group, or do they, quote, never catch up? And that'll give you some sense of the effect of the drug. So I think geriatricians need to get on board with this drug, and here's why. The drug is effective. The drug has risks. We will learn from using it the net compendious benefit of this drug to patients. And the kind of conversations that we're going to have are conversations about, well, is it about lowering amyloid quickly early on and then stopping the drug and following amyloid levels? Is it about a persistent lowering of amyloid that you have to keep people on the drug? Is it about lowering amyloid and also seeing tau go down and following tau? All these things are going to be part of the conversation in the next 24 months, 2-3 years. And this is how I think the thinking about this disease from the perspective of pharmacologic treatment is going to change the way we talk about the disease. And I think our profession needs to be part of that conversation, needs to be talking that language, understanding the way we talk about that language, and contributing our expertise and wisdom to interpreting the data, both to understand its benefits to patients, but also how to help patients understand it and families understand it. Because I think we're uniquely good as a profession with taking care of patients and helping them understand risks, benefits, and balancing those with their other diseases, et cetera. So that's why I'm pro for this drug entering the clinic and for our profession being prescribers for it.
Ken, are you going to prescribe?
Well, first of all, I mean, I don't know. I find the term getting on board. I'm not sure I like that term.
What? It's a train, Ken. Choo choo.
Yeah, I mean, it sounds like, I don't know. I mean, it's sort of like, I don't know, like Alex has been telling me for years, like I should get on board with the Giants and the Warriors. I've been in California 20 years. Why do I keep like rooting for my Chicago teams? I mean, it's like, the problem is doctors,
because every 70 years the Cubs make it to the World Series.
If you wait long enough.
Hope. But it's like we see patients one at a time, though. So it's hard to be. It's hard to be on board like on that level. Let me, here's my thought.
So, Ken, are you going to prescribe it or not?
Well, I recommend it. No, to the average patient. Would I refuse to prescribe it? Like, unlike Aducanumab, where I see it would almost be professionally irresponsible to prescribe it. This is one where I do see room for patient preference here.
Why would you recommend against it, Ken?
So why would I recommend against it? I think the big question is, in the end, is your patient better off or worse off? So I think one of the mistakes people are making here is there's this sense of jubilation here, because for years and years, we've been trying to prove that a drug that somehow involves the amyloid pathway of Alzheimer's disease, that, a pathway that people think but are not sure is the pathway for Alzheimer's disease, it does something in altering the clinical pathology of Alzheimer's disease. And I think for the first time, we actually have a drug that actually shows there is something to that. But I think what's missed is, okay, maybe we finally have crossed that threshold. And this is like, again, hoping like, okay, your sports team has put together a good team and in 20 years they'll win the World Series. Now we've reached that point. But I don't think we have evidence that says this drug is going to make the patient sitting in front of you better, because I think what we have is what you would say a teeny piece of effect that may or may not be noticeable to a patient. And, significant toxicity that and I know we're going to talk about toxicity later, but toxicity that I think we have reason to worry about that is understated, and that the long-term effects of are unknown, where we have good reason for other areas of brain science to worry about. And as even before you get, remember, the whole reason is to make your patient's life better and to help your patients live a good quality of life. And now we're saying okay, just come in for this infusion every 2 weeks.
So Ken,
quality of life to me.
So infusion every 2 weeks, that's a personal choice, as well as obviously thinking through the benefits and the risks, particularly for people who are ApoE4 carriers. But let me jump to very, very simple point. You mentioned patient quality of life. Actually, among the secondary endpoints in the study was the Zaret rating of caregiver burden and the QAL-AD developed by Rebecca Logsdon. And in multiple studies of cholinesterase inhibitors, it's interesting, I've never seen a cholinesterase inhibitor study that showed an effect on the Zaret scale or the QAL-AD. On this study, persons on drug had significantly different scores on Zaret burden scale by their caregiver and patient-rated quality of life on the QAL-AD. patients on drug had higher self-ratings of their quality of life than patients on placebo. And the caregivers of patients on drug had lower scores on the Zarit burden scale than patients on placebo.
So 2 questions on that, because one of the questions from Aducanumab was, did people know what drug they were getting based on potentially side effects, including, needing multiple MRIs for worries about these brain microbleeds? And the second question, well, I'm going to ask that question first. What do we know about how good did they blind patients? Do we know anything?
So in these studies, the question of unblinding obviously is a big one, because the side effects are ones that you can feel. And there'll be periods of time where you sort of stopped your infusion or go on placebo, et cetera. And the bottom line is, when their analyses take into account the potential effect of unblinding, courtesy of side effects. And they did a variety of sensitivity analyses. I don't pretend to be able to sort of understand them all, but I'll take it, and the sense from the reads of the data are that you can't explain these data away by simply unblinding. Yeah. Data on the cognition, data on the functional measures, et cetera, that there is a drug effect here. It's not just a confounding by unblinding.
And the second thought is, and maybe this is the reason why geriatricians should get on board with this drug, is it would be interesting to see what quality of life looks like, not between a group getting placebos twice a week versus this drug twice a week, but a group that's getting this drug twice a week, every 2 weeks, needing to go to the infusion center, versus a group that is not getting it and not having to travel to the infusion center twice a month.
That's a study that would be hard to randomize and blind to. We have historical data,
right?
We have historical data on that. Look, we should probably talk about risks, because the drug does have risks. I mean, it causes microbleeds in the brain, and it causes swelling, edema as well. And those risks are more likely in people who carry one copy of the ApoE4 gene, and they're even more likely in people who have 2 copies of the ApoE4 gene. And so you have this difficult situation of a drug that has well-documented, well-quantified risks, in particular in people who carry a gene that's associated with developing the disease. And the forest plot data showed a dose decrement of response to the drug for each copy of the gene you had. So if you were a homozygote, you had less evidence of benefit than if you were a heterozygote or a non-carrier. My takeaway from that is not that the drug, quote, doesn't benefit homozygotes or heterozygotes. I think that's an overread of the forest plots, but it's pretty suggestive that, and this is going to need some teasing out, that if you are a carrier, you potentially might not benefit as much, and you face a greater chance of these risks. And so I think that does make for a Gordian knot of a very ethically challenged risk-benefit balance that a patient's going to face. Again, my view on that is the patient and their family need to make that decision. Obviously, I'm there. I want to help them make it. I think that's why geriatricians, again, should be part of this, because we're good at talking to our patients and listening. But it does make for a complicated decision. It does. This is not a risk-free drug. And for people who are ApoE4 carriers, it's a particularly problematic decision.
There's some word on the street right now that SI may say recommend against it in homozygous carriers.
If a homozygote patient wants to take it, I would prescribe it, but I think they need to understand that they're going to need careful monitoring for risk, and I may not be able to catch that risk before it becomes symptomatic and causes some problems with gait and falls, et cetera. But again, I would view that as their choice.
And there was also some news about 2 deaths maybe being linked to getting
That's right. There was this patient in this study who died, and then a patient in the open label study who died, both of them in the context of getting ill. And, it's kind of like, it gets a little frustrating when I hear, like, did lecanemab cause their death or not? As we all know in geriatrics, the idea of what causes death in patients with multiple problems, it's quite clear, though, that exposure to lecanemab contributed to their death. I mean, it increases the risk of brain bleeds, and these people are getting blood thinners, one for a stroke and the other for a heart attack. And so yeah, that probably was not a good combination. Yeah. Look, my take on the drug is this drug has real benefit and real risk. And I think the biggest problem I see is after FDA approval, if it gets widely used by clinicians not skilled in how to use it, we're going to see problems with it. So I think actually FDA should put it under a risk evaluation mitigation strategy or a REMS strategy. And a REMS strategy allows FDA to create criteria for who's a prescriber. It allows FDA to set standards for gathering data for risk in a registry, in a database. So it's been used for drugs just like this. There's benefit, but there's also risk. In the early days of MS, it was used for the early MS drugs that could cause PML in addition in the brain, just very similar kind of side effect in some sense. The concern I have is without a REMS, especially in America, wide use by frisky prescribers could cause some real problems here for patients.
Yeah, there's something. I mean, I'm in complete agreement. I'm complete agreement, Jason. That for several reasons. I mean, I think the data registry is super important because if this is approved, we really need to know if the side effect burden is different in a real world practice than in a clinical study, because these are the sort of side effects that, for the reasons you mentioned, are notorious for being worse in real world application. But also, a registry allows us to study some of the long-term, both benefits and risks. Here's one of my concerns, Jason, about risk is that, I'm not, my understanding of some of these risk factors for brain science is that from a lot of spheres, whether brain injury is concussion or brain trauma, you think about like chronic traumatic encephalopathy in NFL players, whether it is like, maybe even psychosocial trauma, like, post-traumatic stress disorder, or even we know from some of the science, I was taught years ago that when you do MRIs on just regular people and you see these little mini strokes that right now have no clinically meaningful impact that we can measure, they do over time. So one of my worries here is we say okay, well, it caused a little bit of brain swelling, it caused a teeny bleed in the brain. Maybe it was asymptomatic. Don't worry your pretty little heads over these problems. I
worry about those. I disagree. When I say geriatricians need to get on board with this drug, that means get on board with using biomarkers to understand what the cause of someone's disease is, in this case, Alzheimer's, and then using MRI technology to better diagnose the disease and look at some of the comorbidities like microhemorrhages. In the case of using this drug, if someone develops microhemorrhages or microedema, you will have to rescan them and follow up, potentially stop drug and or halt it completely if those microhemorrhages and microedema don't clear. So this is not a drug you just give it to someone and see them back in 6 months. Early on, it requires frequent MRIs, and based on those results, even more frequent and or stopping the drug. And I would take that extremely seriously. Yeah.
Yeah, I want to raise another point about disparities in equity. Interested in your thoughts about this. Sharon Bragman wrote a terrific commentary in JAGS where she talked about the need for including persons in clinical trials who are most likely to be affected by the condition. And we know that Alzheimer's disease impacts African Americans and Latinx patients at rates that are 2 times and 1.5 times respectively higher. And that Aducanumab, they had very low numbers of participants from those communities. I think it was 19 Black people. And in this study, I'm interested in how well you think they did. They had 20 Black people in the lecanemab arm and 24 in the placebo arm. In terms of Latinx people, they had 12.5% or 107 in the lecanemab group and 108 in the placebo group. Thoughts about, and there are other equity considerations at play here, right? We have access to, you have to have a PET scan or a beta CSF measure to get in. And then you require frequent infusions. You have to travel to infusion center. You have to have access to infusion center. And then you have to be monitored for these, brain bleeds, brain hemorrhages. So there are other issues in terms of access to this medication if it is approved. Thoughts from the two of you about those issues?
Yeah, I mean, you stated the problem very well, Alex. I'll give you a middle take, as I at least, I have to acknowledge that, the company that sponsored this trial did way, way better than the company that sponsored the Aducanumab trial. There was at least a semblance of taking responsibility for inclusivity. That the minority inclusion was not nearly adequate, but at least it was there and taken seriously. It shows that we have a long, long way to go to make this right. But at least, there was some effort at minority inclusion. At least there was some effort in it. I actually wish they didn't have the age cutoff at 90. There was no reason for that. But okay, they at least went to 90. So, they at least included people who are very old. They included people who had multi-morbidity. So by that, I mean the diseases of aging, beyond dementia. We at least have some data about what happens on people who are on anticoagulants. So that not enough here, but better. Some progress in study design development.
That's the great thing about Aducanumab. It sets such a low bar. Anything.
Yeah.
You got it published in a journal? That's great. How wonderful.
Yeah, no, Biogen set a bar so low that anything other drug company does is automatically virtuous behavior. But I can't tell you how when you chat with the folks in pharma, how much they just roll their eyes at what Biogen did. The challenge I think we face when we think about the need for diversity in clinical trials is diversity with respect to what, for what reason. If it's with respect to the reducing the risk of developing disease and or having disease progression, because these people have already developed the disease in the Clarity study, it's about progression, then I'd want to make sure that your design is going to attend to what you think are those mechanistic factors that might have persons, say, who self-identify as African American progressing faster. I didn't see that going on in this study. Instead, I saw diversity with respect to just equity issues of access to clinical trials, right? That all Americans should have access to a trial, assuming they meet the criteria for entry. And it's odd when as the case with Aducanumab, that there were, you could count on one hand the number of persons of color in the study. Mm-hmm. And I think there are two very dissociable issues with respect to quote, diversity in clinical trials. The former actually is very scientifically challenging because if you think about writing a label, so the label would say this drug is for the treatment of Alzheimer's disease, and here's some other points depending on the person's self-identified race and ethnicity about how you prescribe the drug. I mean, I find that very problematic because behind race and ethnicity are a host of cultural and social constructs. So I'm still struggling with in drug development, thinking about these issues with respect to understanding mechanism or prescribing. But in terms of access to research and its benefits as well as its risks, it's a shame that our studies tend to simply reflect only a small slice of America. Absolutely.
Well, let me ask a follow-up question real quick. When I look at the supplement, it seems like, again, caveat is that you look at those forest plots, it looks like this mainly works for white men who are ApoE non-carriers.
Yeah, so this is the issue with forest plots, and it's kind of funny. Well, first of all, you get power issues. Yeah. Okay,
it's not designed for me to make that statement, right?
Right. But secondly, forest plots, it's supposed to look at like overall, am I seeing a trend that favors drug, or overall, am I seeing a trend that favors placebo, or am I seeing things all over the place? And the forest plots are pretty compelling for a trend that favors drug. Having said that, I'm the one that fixated on the ApoE because of the dose response aspect of that. 0, 1, 2 copies of ApoE4, you see across all the endpoints the same dose response reduction in benefit. That doesn't lead me to say it doesn't work for people who have ApoE4. It makes me concerned, and we need to understand more what is going on here. The ApoE4 carriers may not be as good responders. But I wouldn't look at those forest plots and say a woman from Europe can't benefit from this drug, because that's what the plots would suggest. I would just, at some point you're getting into issues of multiple comparisons. Also, biological plausibility. I mean, can someone limn up some scientific explanation for why Europeans aren't responding? The answer is, I think it's like we could have done astral signs and shown that Sagittariuses don't respond to the drug. I mean, I think we have to be very careful.
All right, I got 2 more questions. I know we're running out of time. Ken, what do you think about this question? Do you think this study is proof that the amyloid hypothesis is correct, or do you think that based on these very small changes, this is proof that there's a lot more going on besides amyloid and Alzheimer's disease?
Well, you astutely framed the question correctly by not making it an extreme question, because I think the answer is, for the first time, we actually have evidence that there may be something to the amyloid hypothesis, that when you change amyloid in a big way, you do something to the clinical parameters of the disease. I think, on the other hand, it's also very convincing proof that there's probably a lot more there. That this study, no one will disagree, like as Jason noted, this changes amyloid in a big way. It's like, it's a great win. This like obliterates amyloid. But, if it was all amyloid, you would have expected, like, why didn't the progression of disease stop? So remember what we're talking about in these 2 groups. Both groups get worse. One group gets worse less. And that's important, getting worse less.
Yeah. I mean, Ken's points are well taken. I think geriatricians need to get on board with this drug, because this is the beginning, not the end, of understanding what happens to a human's brain when you reduce amyloid. And my hunch is 5 years from now, we're going to be back on this podcast talking about the various subtypes of Alzheimer's disease that do and don't respond to the various drugs based on a variety of factors. And you know what? That's been the story in a variety of complex chronic diseases, especially associated with aging. But, the effect in this trial, I believe, is a real effect. This is not a statistical artifact. I think it crosses thresholds such that it's effective. It's got risks. That's the kind of drug that can be in clinical practice. I'd like to see my colleagues in our field, because I think geriatricians are the kind of doctors to diagnose and treat people with these diseases. And I hope that our field recognizes that this biomarker transformation of the disease is something that we can be part of and inform and improve the way these diseases are diagnosed and treated, rather than be on the outside saying there's not enough benefit, or amyloid isn't the only cause, so I'm not going to pay attention to these data. I just don't think that that's clinically responsible and scientifically responsible for us anymore as a field.
Can I follow up with that? And just to be clear, are you suggesting that geriatricians should set up infusion centers and be prepared to send patients for, screen patients with PET scans, or do CSF?
Screening, no. I think geriatricians should be prepared. Geriatricians should be in their practice for patients who have MCI or mild stage dementia that looks like the Alzheimer's phenotype, to be comfortable with ordering biomarker tests, particularly amyloid, to confirm the person has amyloid, likely therefore has Alzheimer's, and engage in a conversation about this drug. I think they should be comfortable with. I don't have to have an infusion center, but following up the next steps in terms of MRI imaging, et cetera. I think that these drugs are probably to be prescribed in what we're very good at, multidisciplinary teams. Where once the drug is started, our colleagues in our practice can help with monitoring response, such as paraprofessionals. But yeah, I think we should be comfortable being prescribers for these drugs in a responsible and reasonable way. Absolutely. Yeah.
So Ken, I'm going to ask you the same question, but I want you to think about this. Geriatricians are generally one of the highest satisfaction fields, but the lowest paid fields. Set up an infusion center. The average salary for geriatricians all of a sudden skyrockets. Should geriatricians get on board?
Well, first of all, am I released from the debate stance, and can I say what I think now?
Yes, you can say what you really think.
Okay. That's an economic issue for our field. I honestly don't care if we're the infusion center or we send our patients to the memory and aging center. I do agree with Jason that there's a lot more to learn, and our field should be involved in learning about it. What I would say is my take on this is my instinct is that for the average patient with MCI and mild Alzheimer's disease, this probably will not be worthwhile. But I agree with Jason is that the evidence is such that I owe my patients a serious discussion, and I would ask a lot of questions about their values. I would, what's important to them, what would matter to them. I would actually ask them like how much, the protocol would disrupt their life. And if they say they don't care about that protocol of having to go to a clinic every 2 weeks, that might influence what I recommend. I'd be open to them that reasonable people do see use in this drug. Mm-hmm. And if I had a patient after that discussion who wanted that drug, I would facilitate their access to it if that was their decision after, hearing a full discussion.
Let me ask you the last question. Given that it doesn't seem like the decision on the FDA, how they decide things, does not seem that they take much into account besides the whim of the director. Sorry, I'll just get off my high horse there. Should the FDA approve this drug? Jason, I'm going to turn to you first. Ken, you can think about it. Jason, I heard with maybe yes, but maybe thinking about...
No, I think FDA should approve this drug, full approval. I think we should stop this conditional approval nonsense. It needs review for full approval, and I think based on the data out there, it should get full approval. I think FDA should approve it, though, with a risk evaluation and mitigation strategy attached to it, or REMS.
Okay. Ken, what do you think?
Yeah, I would just sort of say, I have a number of requests. So like one of the requests I have is I know there's this little box in the study that asked if the authors will make the database available, and it's checked off no. That's totally unacceptable. So that this database needs to be open for the academic world and the policy world to look at. I would say probably with a very rigorous risk evaluation mechanism that it needs to be done and we need to assure that it's approved in a safe manner, that patients are properly informed about the risks and benefits, and that anyone getting the drug be entered into a registry where we really understand it. We can't wait 10 years. If we find the rate of hemorrhage was 3% in a study and is 20% in real life practice, we need to know that soon. We can't wait to learn that.
Wow. Ken Covinsky agreeing that the FDA should approve. Times are a-changing.
We won't get fooled again, right, Ken?
No, yeah, with reluctance.
It's like the Oscars. Alex is starting to play in the background.
The line it is drawn, the curse it is cast. The slow one now will later be fast, and the present now will later be past. The order is rapidly fading, and the first one now will later be last, for the times they are a-changing.
Jason, Ken, thanks for joining us on this podcast. That was absolutely fabulous. I learned a lot, as I always do from both of you.
Well, thanks a lot, man. Really enjoyed it. Good to be here.
You. Fun being with you and good seeing you, Jason.
And to all of our listeners, thank you for joining us on this Jerry Pal podcast. And a special thank you to all of our listeners who've donated $250 or more to the Jerry Pal podcast,
including Meg Walhagen, Thomas Quinn, Rochelle Bernacki, Louise Aronson, James Tulsky, Arden O'Donnell, Mike Steinman, Marianne Forcia, Ashok Krishnaswamy, Nancy Lundeberg, Gail Cooney, David Schiffeling, Cheryl Phillips, Jessica Ng, Harry Hahn, Elizabeth Chung, Kathy Foley, Lloyd Woolstat, Christine Ritchie, Matthew Schuster, Daryl Owens, Susan Nelson, Christopher Heck, Lindsay Yorman, Mo Rizawi, Sue Borson. Thank you very much.